Thanks for hosting me. Thanks for the invitation. As we were just discussing before the start, I think this invitation was originally extended about a year ago, and so it's great that we've finally got to this point. I realized also when I was thinking about what I would talk about today that a year ago, I think the content of my presentation would have been quite different. Quite different in the sense that it would have focused almost entirely on the research, and that's still going to be the case today, except that today I'm going to try to position, at least at the start of my talk, some thoughts or present some thoughts on why doing research in a disease endemic setting is important, why investment in infrastructure, and more importantly, people to do the research in endemic settings is important. So to everyone out there who has seen any of my recent presentations of our work on Mycobacterium tuberculosis transmission, I apologize in advance because some of the work will be familiar to you, but I do hope that I present it in a different way and at least try to encapsulate some of what was promised in the abstract for my talk. So my title is Collapsing Boundaries in Infectious Disease Research. And I thought initially just to orientate everyone who is not from this setting as to the location of the Institute and its composition. So for people who haven't been here, anyone who's online, the ICGB, as Louise mentioned, is actually located in this building here on the right-hand side of the photograph. Everyone associates the Institute of Infectious Disease and Molecular Medicine with this glass building, but in fact the Institute extends across 5 individual buildings. It was established over 20 years ago with a clear vision to be an international center of excellence tackling diseases of major importance of Africa. It has 4 core mission statements to conduct basic clinical and public health research that is leading edge and relevant. It intends to develop indigenous scientific capacity to influence health policy and practice and to build partnerships. And I think, again, thinking about where I would have been a year ago, where there's been a lot of discussion over the last year of how we increase our partnership with ICGEB as a major component or a co-group within our precinct. And so to build partnerships and to underpin everything with the values of excellence, collaboration, and transformation. The IDM is really comprised of multiple member groups, many of which have global reputations in their own right. And I've encapsulated several of their logos there on the right-hand side. So we are a trans-faculty postgraduate research institute, which draws our membership primarily from the Faculty of Health Sciences at the University of Cape Town, but also other faculties. And we have adjunct and affiliate members from elsewhere. We're just busy compiling our annual report for 2024 and just pulling out some of those highlights. We host about 30 research groupings, multiple members at different stages, full members, associate members, and an exciting cohort of emerging researchers who are our IDM fellows. We're a postgraduate and postdoctoral research hub. We host a large number of clinical and professional staff. We also host a large number of PhD students. We generate a lot of papers. We host and generate original research which gets patented, and we try to meet our commitment to global collaboration by extending our collaborations each year. And so for 2024, there are over 156 individual global collaborations. The Institute is built around core platforms. I don't like to use the word facility, much more platforms. They're collaborative areas for members and expert staff to drive the most advanced research. We're best known, I think, originally for tuberculosis research and HIV research. So we have BSL-3 containment labs, and increasingly an expanding cohort of advanced analytical capacity, proteomics, flow cytometry, and FACS, light microscopy. We are underpinned by large bioinformatics and computational biology groups, as I'll highlight later. We have an histology core, a recently launched microbial interactions lab. We've got a translation hub in development and a genomics hub that's planned for the future. And those cores then support the enterprise, the research of all our member groups. We are, as the IDM, the basic science platform in the only discovery research platform that is Wellcome funded and exists outside of the United Kingdom. And as the basic science platform, we fit into a landscape which allows continuation of research from basic science through data integration and clinical research. So therefore, thereby filling the full continuum of research that can go from bench to bedside and bedside to bench and back. We have members who work on drug discovery and development, such as Kelly Chibali's H3D Center. We have groups which are working on tuberculosis vaccine and testing, as well as TB drug trials. We are active outside of just TB and HIV, so growing footprint in mycology, established groupings in virology and helminth research. We support group leaders in genomics and precision medicine, and these are in the form of 2 South African Medical Research Council extramural units. As I mentioned already, we have a large bioinformatic and computational biology platform within the Institute, and host Professor Nikki Mulder and H3 Africa, and are excited to be associated with the newly launched African Bioinformatics Institute. We also are expanding into different areas, and so through recent attraction of researchers, we have established strong links with the Forensic Pathology Institute, which is at the Groote Schuur Hospital just across the car park from those who know this location. And this is really to drive research in post-mortem studies. We're not just about research. The IDM supports groups which are driven to translate research findings into products, diagnostics, drugs, vaccines, etc., that can be really change people's lives. And an example I've captured here is the genital inflammation test for HIV prevention, or GIFT, which has been led by Professor Joanne Passmore and her team. We also like to think of ourselves as a really important hub for developing the next cohort of emerging researchers, and through partnerships such as with the CRIC Africa network and LifeArc, we host 2 recent African Career Acceleration Fellowships, Ruan Murray and Munyaradzi Mosfosfi, both of whom are working on projects which will lead to products for implementation. Within the IDM, we host groups and entities which are trying to make a major difference across the continent. One of the more exciting and more recent of these is the Africa Microscopy Initiative, which was launched just over 2 years ago, which essentially functions as an open access light microscopy facility for the continent, with multiple very advanced light microscopes available for use by African researchers from across the continent at no cost, and actually with facilitated access into the center. And then within our research groups and through certain dedicated entities such as EWOSA, we try to impact the communities in which we operate. So that gives a quick overview of the IDM as an institute. And I thought, as I said, it was important to do that because it's within that context that some of the work that I will present now, some of the actual research, has been conducted. And there'll be a couple of themes which I'll try to pick up as I move through the research and conclude at the end. But one of the most important of which is the ability to attract multi-disciplinary expertise in a single space in order to drive research, local research, which actually I would argue in many cases can't be done anywhere else. And that this is why it's so important to invest in people and infrastructure and in the preservation and ultimately the growth of entities such as the IDM and ICGB in this context to continue to deliver advanced research which can impact local health. So I put this meme up, which I quite like. There have been several along these lines recently, which I've come across, which argue for basically the fact that people overstate or overpromise in their paper abstracts or poster abstracts and titles, and in this case, maybe even the talk abstract and title, and that the what's promised doesn't come close to what's delivered. I hope in my case that isn't the criticism that's leveled, and I'll try to justify now the second component, which I captured in my talk abstract, which was really to try to highlight how a shift in thinking about, in particular, bacteria as infectious agents is changing the way that we tackle infectious disease such as tuberculosis. And I like to think that some of the work that I present now has contributed to at least reinforcing some of the key insights and key themes that have been emerging over the last decade or so. So for those who might not operate in bacterial pathogenesis sphere or don't read this kind of literature a lot, I thought I would just signal a comment which was published just over a decade ago by Arturo Casadeval and Lisa Ann Porofsky in Nature. It was quite provocatively titled, "It's Time to Ditch the Term Pathogen." And I've captured in the red box really the essence of the comment, which is that disease is as much about the host as it is the infectious agent, and that the focus on the microbe alone is hindering research into treatment, say the authors. This comment actually came on the back of several years of work by the same authors, one image of which is captured here from a 2023 paper, where they had developed a framework called the damage response framework of microbial pathogenesis for trying to understand bacterial disease as not just being about bacteria as pathogens and bacteria as virulent entities which cause disease, but rather to recognize that pathogenesis is an outcome of an interaction between a host and a microorganism. And that, very importantly, the second fundamental tenet of this framework is that we are focused on the host relevant outcome of this interaction being disease, and that disease is really a measure of the amount of damage to the host. And so what's captured in this cartoon or figure, which is partially quantitative, but more qualitative, is that there's a measure of host damage and potentially even host benefit below the x-axis, and that the x-axis is divided into the host response, wherein the parabola in this case highlights an interaction with an organism where an absent or weak immune response leads to host damage, but by the same token, an overly robust or excessive host response may lead to disease or ultimately even to death. And on the right-hand y-axis is really just different stages of disease, moving from commensalism, colonization, latency, disease, which occurs at some kind of threshold, which would be pathology or presentation at clinic, and then the worst or the ultimate outcome being death. And very importantly, in terms of this framework, the third element is that host damage can result from either microbial factors or the host response. So, for example, where the immune response is overly robust, or a combination of both. And so it recognizes that bacterial or microorganism attributed disease is a much more complex entity than simply the case of a failed immune system, for example, or a hypervirulent bacterium. Certainly those factors influence the outcome, but they don't determine the outcome. More recently, Arturo Casadevil, in an interview which I would recommend reading on the American Society for Microbiology website, summarized this in a single epithet, which I really like, which is that pathogenesis is not a trait of an individual organism, it's an outcome. And so where do we see examples of that? Well, in our setting, I'm a mycobacteriologist, as Luis highlighted. I work on Mycobacterium tuberculosis as the bacterial cause of tuberculosis. And I work in a setting here in South Africa, which is consistently ranked amongst the highest TB incidence regions in the globe. So this is a figure from the 2024 Global TB Report highlighting incidence rates for well over between 300 and above TB cases per 100,000 population per year. So you can see the darker the green color, the more severe the TB incidence. We also know that in South Africa, we actually sit at the nexus of three converging epidemics. That's drug-susceptible TB, drug-resistant TB, and TB HIV. And although these figures change somewhat, or whether they're represented each year in the WHO report, we are consistently a member of this unfortunate group of countries which have high rates of all three of these disease entities. During COVID, the first national TB prevalence survey was conducted in South America. It was published shortly thereafter. And I'm just going to draw your attention to two key figures which emerged from that survey. So just looking at different age groups, 15 to 24-year-old, 25 to 34, etc., and then the over 65s. What is highlighted here is where is a measure of the prevalence, the TB prevalence, to a case notification ratio. And in both of those age groups, 15 to 24 and over 65, it approaches 3, which means that actual TB is three times more prevalent than the case notification rate. And across all the different age groups, that ratio is above 1, meaning that there's more tuberculosis in our communities that is being detected. If we think about tuberculosis as a kind of a disease cycle, we often see figures in the literature which look something like this. An active TB case produces organisms often assumed to be via cough, which are transmitted to an uninfected individual, and that person may go on to become TB infected with or without disease, which may be subclinical disease, largely defined as being TB without clear symptoms, or active TB disease, and thereby completing the disease cycle, with the potential that there could be transmission during the asymptomatic disease state. How this is studied, though, and this is one representation of it, is that in general, research groups separate their research into studying the organism specifically, which are most of the components here, which are highlighted with the blue arrows. So a lot of in vitro work with Mycobacterium tuberculosis in a fundamental microbiological model, or in host-focused studies looking at different animal models or ex vivo models of infection, or in some cases using clinical materials such as blood, urine, biopsies, or more recently post-mortem studies. By far, the predominance of TB work is done with sputum as the source of the organism, and then subsequent laboratory culture. What this highlights, though, is that there's a siloing of research. It's often very difficult, practically and scientifically, to bring teams together to really work on tuberculosis as a system, as an interaction between the bacterium and the host, and between that bacterium and the host in a specific environment. And it was this kind of thinking which, over a decade ago, working with my now close colleague, Professor Robin Wood, and several other members of our research team, we started to address the problem, or we thought we would try to address the problem of understanding where Mycobacterium tuberculosis was coming from. So where is the organ, who's transmitting the organism, when during disease stage, or as a function of their disease state, are they transmitting the organism, and how quickly does tuberculosis treatment impact the release and transmission of Mycobacterium tuberculosis. The reason for our interest was that an analysis that Robin and colleagues had done before this, they had looked at TB rates, TB notification rates in major cities across the world, including Cape Town, and had identified a really stark trend or pattern. So what was clear is that in global north settings such as New York, each decade over the last 100 years, there has been a steady decline in TB notification rates. I should have pointed out at the start was that their selection of cities was based on cities where there were really good records of TB rates going back over a century, and actually Cape Town is included in that number. What's clear when you look at this image is the contrast between New York and Cape Town, where over the same 100-year period, Cape Town has essentially remained or returned the same rates, even showing an increase as a consequence of HIV in the early 1990s and thereafter, and then an expected increase now just post-COVID. Another important thing to highlight in this image is that the decline in TB incidence in New York preceded the introduction of antibiotic chemotherapy. So what brought tuberculosis down is not the availability and the utilization of anti-TB antibiotics. There's other factors which allowed that decline to start happening, and it didn't happen here in Cape Town. And it didn't happen in Cape Town despite the fact that over the same 100 years from Robin and colleagues' analysis, the TB control measures which have been implemented in Cape Town have been done so as rapidly as they were done internationally and in other settings. And in some cases, Cape Town even took the lead globally in the then most current TB intervention. So despite the implementing best practice for TB control, the rates in Cape Town have remained consistently, of tuberculosis have remained consistently high. And the question is, why? What are those factors? And for us, a key question was, to what extent is TB transmission a major source of these elevated rates? And is there the potential that there might be individuals who are super spreaders, for example, or are some of our assumptions about how tuberculosis is released and spread might be erroneous or misinformed? So in order to address this, the work that I'll present is all conducted at the Aerobiology Research Center on the Desmond Tutu Health Foundation's Masiphumelele clinical site just outside Cape Town. And reinforcing a theme that I mentioned earlier, the team that tackled this problem is a multidisciplinary team. It's microbiologists, clinicians, engineers, computational fluid dynamicists, research nurses, community health workers, students, bioinformaticians, etc. All of those expertise captured in this single photograph, which doesn't actually reflect the full staffing of the site. The research site is Masiphumelele, which is a low-income, high-density um, a township just outside Cape Town. The area is actually physically constrained on three sides, naturally by the road and by a road and a wetland, and this has resulted in a fairly stable population over the last couple of decades. It's estimated at around 23 to 25,000 individuals now, and because of the close working relationship of the Desmond Tutu Health Foundation with the community, a lot of the work which I'll describe in subsequent slides has been enabled through good interactions and also the ability to map individual locations within this area, as I'll highlight towards the end. So when we started out, an accusation or a concern leveled at our intention as a team, which included microbiologists, of identifying individuals who are producing Mycobacterium tuberculosis by actually finding the organism, was that we were in effect hunting the boson, an extremely rare entity which even if it was there, we might not be able to detect, owing to the difficulties in sampling and the difficulties practically in handling extremely small numbers of organisms. Robin, in his genius, imagined that we would develop something like a TARDIS for those who are Doctor Who fans, a box which could actually travel in time, potentially along with the bacillus, that we could use to understand the actual journey that the organism was taking as it went from an tuberculosis infected individual to a naive or a new recipient. The TARDIS, or the first version, actually looked like this. And so for those who are familiar with TB work will recognize this as a sputum collection booth. In this case, it's been heavily modified by the addition of all sorts of additional analytical equipment and air filtration systems, so that ultimately what was produced was a personal clean room, which would allow us to sample all of the air produced by an individual sitting in the booth for a given period of time. Critically for us, when we set out to do this work over a decade ago, we were determined that we wouldn't be dependent solely on cough. So we weren't interested in just getting potential MTB carriers or MTB patients to cough in order to produce organisms. We wanted to understand the potential that organisms might be produced in a cough-independent manner. Hence the design of this booth, which allowed capture and filtration and concentration of all the air produced by an individual sitting in that booth. We were very excited early on when taking air from the booth, concentrating it and depositing it on aluminium foils and putting those foils under an electron microscope, we saw structures which looked a lot like potential bacilli. We were very excited. The reviewers of the first papers were less convinced and said they may look like rod-shaped organisms and therefore may potentially be M. tuberculosis, but we need greater proof, and that was a fair criticism. We shifted our collection mechanism as methodology as a consequence of that criticism. So instead of taking air and putting it onto grids, which could be examined by the electron microscope, we passed air through a impactor. This is an Anderson multi-stage impactor. Essentially what it does is separate particles in air streams according to size and then deposit them onto each one of these individual stages. And for microbiologists, each of the stage can contain a petri dish with a solid growth medium. So if you can pass air through the impactor and capture organisms and then incubate the individual plates from each of these stages over time for M. tuberculosis, 4 to 6 weeks later, you can identify colonies on the individual plates. You can take those colonies, extract the DNA, and you can sequence the DNA. And that's what we did. And so from confirmed TB patients sitting in the booth, just capturing their air and passing across the stage, we were able to identify and confirm the presence of Mycobacterium tuberculosis in individual colonies. For us as microbiologists, that was exciting, but it was unsatisfactory because we were only seeing colonies on plates, and actually what we wanted to see was individual organisms. And so through a lot of engineering over the last decade, that original tar disk developed into what now exists in the site, which is the respiratory aerosol sampling chamber, which is shown here. So this is a now transparent version of the collection booth. You can see a participant sitting inside the booth, breathing into a funnel. This enhances our collection and allows us to reduce the collection time. It's not essential, so we can do it without the funnel. Again, importantly, what the system does is it allows us to collect aerosols without requiring cough. And on the back of this system is a collection device, which is a liquid cyclone, effectively a liquid vacuum cleaner, which pulls through the air and collects all of the air, the material in the air, into about 15 mils of liquid. There are many different other systems that are built into the aerosol sampling chamber, which allow us to monitor the status. We can measure carbon dioxide production, look at relative humidity. The air is multiply filtered to ensure that it's clean from one participant to the next, and we run an empty booth control between between each participant. And it's fitted with an ozone decontamination system, so that we can decontaminate between individuals. What I should have highlighted on the previous slide, you can't really see, is there's a hatch just over this person's shoulders, which leads into the BSL-3 lab. So samples are collected here from a patient, collected into the liquid cyclone, and then passed through the hatch into the biological safety level 3 lab for further processing. That further processing involves two key steps. One is a microscopic identification of potential MTB organisms, and for this we use fluorescence, a fluorescent probe. It's a DMN dye, which is linked to trehalose, which is a major sugar component of the mycomembrane. So mycobacteria take up the trehalose through an active enzymatic process, which is catalyzed by the antigen 85 mycolyl transferase complex. The trehalose is pulled into the cell wall of the organism, or the mycomembrane, and when that trehalose is a fluorescently tagged trehalose, it means that incorporation into the cell wall creates a fluorescent signal, which allows us to see the organisms. So we can detect fluorescent bacteria, which distinguishes them from the rest of the background signal. Importantly, I'll highlight here that this probe is not specific to Mycobacterium tuberculosis. It's restricted to a group of organisms which possess an antigen 85 mycolyl transferase homolog. So other organisms have the ability to take up trehalose, will fluoresce in the system. And as a consequence of that, we spent a lot of time several years ago, and we continue to develop these algorithms differentiating Mycobacterium tuberculosis based on the incorporation profile of the probe from other closely related mycobacteria. This is a non-pathogenic mycobacterium, and then other non-mycobacterial strains such as Corynebacteria, which also take up the probe. And through this, we've developed basically decision trees, machine learning methods that can allow us to, with a high degree of sensitivity and specificity, distinguish M. tuberculosis organisms from non-TB organisms. And examples are shown here. And importantly, the sensitivity of the microscopy means that we can capture high content, high resolution data For example, fluorescence intensities across individual cells, which we can then use to develop sort of consensus morphological profiles of aerosol-released organisms. Our fluorescence system is coupled to a molecular detection of Mycobacterium tuberculosis. And so for this, we use a droplet digital PCR based assay, which identifies the RD9 or region of deletion 9 deletion, which differentiates Mycobacterium tuberculosis from all other mycobacterial descendants of the last common MTB complex ancestors. So we can get with some confidence a molecular confirmation that our samples contain MTB and not other mycobacterial species. And what's highlighted here is the correlation between our detection of RD9, so this is our MTB specific molecular signal, with the DMN trailers fluorescence signal. And so utilizing that combination of concentration in the respiratory aerosol sample chamber, labeling with DMN trailers, and molecular detection of the M tuberculosis specific genome, we can generate the data which I'll present now. And utilizing this over the last few years, we put out a number of studies describing this work and our constant enhancements and adaptations of our protocols for improving our ability to capture and detect individual organisms from individual TB patients. And so some representative images are captured here. So what have we shown? And this really gets to the important part. And I see that I've got about, I think, 10 to 15 minutes to go. I hope that's right. So a couple of years ago, one of the first things we did is to say, well, we've built a system now which allows us to capture individual organisms at potentially extremely low number from individuals who've been identified as MTB patients. Let's start to test some of our hypotheses. Let's also start to test some of the dogma which exist in the field. And so this study was published 3 years ago, where we looked at 3 different maneuvers which were performed by confirmed TB patients. So these are individuals who've been through the clinical screening and have been identified as TB patients, and therefore are due to start anti-TB chemotherapy. But we have a window before they start the chemotherapy in which we could capture or sample their aerosol production. And the data which I'm going to highlight here, it's published, I won't spend a lot of time on it, reflect 3 different maneuvers: a forced vital capacity, which is a artificial maneuver designed to generate bioaerosols according to the bronchial fluid film burst model. In some ways, basically being the mechanism that we use to try and generate as many aerosols as possible. TIBR is tidal breathing, so that's just normal breathing as an individual sits inside the booth. And then cough is when individuals are specifically requested to cough, and you can see those manifest as individual spikes, a bit like the forced vital capacity. So using these 3 different maneuvers, we were able to demonstrate that cough produces more M. tuberculosis organisms per maneuver than either of the other 2 respiratory maneuvers, tidal breathing or forced vital capacity. But importantly, we were finding organisms during just normal, regular breathing by confirmed TB patients. And when we extrapolated that, we realized that individuals breathe well over 20,000 times a day, whereas coughs, even in the most severe TB patient's case, tend to maximize at around 500 coughs per day. And so this suggested the potential that tidal breathing might be a major source of the release of organisms in our setting by confirmed TB patients. That result allowed us to move into a different clinical trial where we wanted to now look at whether TB treatment reduced the production of organisms and how rapidly that occurred. And so this study, which we refer to, can be a bit confusing. We refer to it as the ABC study. It's ABC because our group A's are confirmed TB patients. So these are individuals who are gene expert positive, who are identified at clinic, and who are due to start anti-TB treatment. The group B, which is the B in the ABC, these are individuals who are gene expert negative, but are identified as likely TB patients based on clinical criteria such as chest X-ray, and so are given a clinical diagnosis of tuberculosis and are also due to start treatment. And then our control group for the study, the C, were those who presented at clinic, who self-presented, so felt sick, but were gene expert negative and failed the clinical criteria for a TB diagnosis, so were not diagnosed as TB, and so were not intended to start TB treatment. So what's reflected here is if we looked at the A, Bs, and Cs, you can see a much higher degree of symptoms as expected in A's, slightly less in B's, and slightly fewer in C's. No other major differences between the groups. And we were very surprised at our just very first measures when we were identifying individual MTB organisms, not only in our group A participants, so these are the confirmed TB cases, but also in the group B's, and most importantly in the group C. So this is at time zero, at initial diagnosis. And so we're finding organisms across all three of these. So this threw out for us our study, because it meant that the C, the control, was no longer a control. We were finding just as many organisms in C's as we were in B's and A's. And the consequence of that was that rather than this being a single time point study for the C's, we then had to follow up the group C's just as long as we did A's and B's, although C's never went on to anti-TB treatment. Only A's and B's did, because C's had not been given a TB diagnosis. We observed over time a reduction in symptoms across A's, B's, and C's. It appeared to be slightly more rapid in C's, although this wasn't really significant. But this brought about the next major surprise, is that, and what's shown here is visit 1, that's baseline, 2 weeks, 2 months, and 6 months. So this is on completion of anti-TB chemotherapy. We were finding organisms in group A's and B's, so these are individuals who've completed therapy. We're still finding aerosol production of MTB bacilli. And so that was the first major finding. The second one was that we see a similar decline in MTB production across A's, B's, and C's, even though it's only A's and B's who were on treatment. So how do we explain this result? Well, looking in the modern literature, a very highly cited recent paper from about 5 or 6 years ago, postulated that there might be different disease dynamics at play with M. tuberculosis. In particular, this notion of a group of individuals who might cycle in and out of symptomatic or TB disease and then non-disease state. So essentially self-clearing the organism and the disease. And actually that notion has historical precedence. So this is just under 50 years ago. It was postulated by a person working in India. Again, this notion of there might be cycling of individuals in the community. And so what we postulate at this point, we don't have direct evidence of, is that for the group C's who are presenting at clinic because they're not feeling well, but who don't meet the classification of TB disease and so don't go into treatment, these may be individuals who are able to cycle in and out of a symptomatic to asymptomatic state. Of course, this raises questions about the potential that these are individuals who might be contributing significantly to M. tuberculosis transmission in the communities. This study generated a very generous and insightful and quite provocative commentary by Sarah Fortune from Harvard, basically saying, should we worry about the bummock? And so what she was arguing is that we, in most TB literature, think only about confirmed TB cases, and so that's the tip of the iceberg, the visible part. But our studies such as this ABC aerobiology study revealing the presence of a much larger lurking threat beneath the surface, and that's the bummock of the iceberg. So we went back into our community in order to test this. So I've shown you an aerial shot of Massachusetts. A benefit of long-standing association with community is that each of the individual plots of land or earths contains a household, and those households can number anything between 2 to 15, 20 individuals. We have a digitized map of land and the land distribution within the community. And so we could run a randomized community study where each day the computer spits out an earth number, and the recruitment team would go out and invite people to come and participate in a study where they were asked to produce bioaerosol for our analysis for M. tuberculosis production. So this is just sampling from the community with no pre-selection for symptoms or disease diagnosis. And the study was published just last year. It was broken up into 2 cohorts, essentially a point prevalence study, which numbered 39 individuals who were sampled at a single time point, so were recruited and sampled at that time point, again with gene expert measured and interferon gamma release assay, which gives us a measure of an immune recognition of M. tuberculosis. And in the second cohort of 50 individuals were followed up at 2 weeks and 2 months for subsequent visits where the same samples were collected. And so again, what really surprised us, this is going out into the community. At baseline, we note that about 80% of our participants produced one M. tuberculosis positive sample. Again, we're sampling forced vital capacity as a maneuver, tidal breathing, and cough. And this was just the baseline measure. When we looked at the longitudinal release in the second cohort, we were also struck by the fact that the rate of bioaerosol positivity through time sat at between 80 and 90%. So at each time point, we're identifying about 80% of individuals sampled are producing at least one sample which contains viable M. tuberculosis organisms. When we look at individuals and track them over time, we can conclude that about 70 to 75% of the community are low, constant producers of MTB organisms. And there was only a single individual who across all 6 samplings, so it's 3 visits with 2 samplings at each visit, baseline 2 weeks, 2 months. There was a single individual who consistently returned an MTB negative sample. So high proportion of very low level MTB production in the community. When we represent the data in this way, I think it becomes, I like to think, quite convincing. So if we go back to our ABC study and we look at the MTB counts at each of the visits, visit 1, visit 2, visit 3, what we see over time is a decline in organism number. And remembering A's and B's are on TB treatment, whereas group C gave us that surprising result of apparent self-containment of MTB production. By contrast, when we look at group E's, just those who are randomly sampled from the community, we see no steady decline over time. We just see a constant, sustained low level of MTB production. So now in conclusion, as we look at these data, we set out to, with this hypothetical map, that we might find some active TB cases in the community who had been missed. We might find MTB infected, basically as bioaerosol positive individuals. We might find some subclinical TB cases, and we might find uninfected individuals. And what we were able from these two studies to do is actually now to say that there's around about 20% of our population are bioaerosol negative. If we look at their quantiferon results or the interferon gamma release assay results in those individuals, it's about 50% who are positive and 50 negative. In around 80% of the population, we are finding individuals who are producing MTB bioaerosols, and so about 4% of the population are actually satisfy the criteria of subclinical TB. So these are gene expert positive individuals who are also bioaerosol positive. And again, the quantiferon positive to quantiferon negative ratios across all of these individual subcategories is about 1-to-1 or 50%. So there's some limitations here. First of all, we are using Diamond-Trailo's fluorescence as our inclusion criterion. So we're only counting fluorescent organisms. We're not sure of organisms that we might be missing. We're applying very strict morphological criteria, whereas throughout the bacterial literature and the historical M. tuberculosis literature, there are lots of reports that organisms might have much more morphological plasticity, but we're looking for very defined bacillary shapes. We are still incapable of generating or capturing molecular data from single organisms. This is something which we really need to work on in order to get clear orthogonal data from an imaged organism. It's fluorescent positive. This is its genotype. Ideally, this is its RNA transcriptome or proteome or lipidome, etc. And very importantly, we don't have a measure of infectiousness. So we're finding organisms, but we don't know their inherent capacity to go on and infect and cause disease in the next recipient. And so a number of questions emerge from this. What is the anatomical source of the aerosol bacilli in these asymptomatic individuals? Can we start to think about MTB colonization? I use this word very cautiously. And is this the default in high burden settings? Is it potentially protective? And if so, what are we potentially doing with a TB vaccine? And then to come back to the theme of real theme of my talk is, what does this mean for our understanding of Mycobacterium tuberculosis as an obligate human pathogen, the way that I was trained and the way so many of us were trained. And so if we look globally, we know that there's unequal distribution of M. tuberculosis across the map, with particular high burden settings, including South Africa. Even within those settings, there are unequal distribution. So we don't find a lot of tuberculosis in affluent, low, high resource, low density settings, whereas just across, say, a railway track or a road, we might find high tuberculosis in a much more high dense, crowded, low resource setting. What is it that differentiates these two settings and causes high rates of tuberculosis in one versus the other? So this comes back to that question that or that epithet that I raised earlier from Arturo Casadevall. Pathogenesis is not a trait, it's an outcome. And being able to do work like this in our setting and identifying that potentially Mycobacterium tuberculosis is much more common in communities than we thought. And as a consequence, or a corollary of that, that tuberculosis as an outcome is a relatively rare outcome. How can that enable us to really understand the true causes of MTB, productive MTB infection and disease? A lot of people have been involved in this work. I haven't given nearly as much credit as I should have to everyone who has worked on this project over 10 years. Many names are captured here, many funders are captured here too. But I think that we're probably just out of time. So sorry, I'll stop there.